So today we are going to discuss simple staining. Simple staining is one of the staining technique. Most of the microbes are colorless, small, and difficult to see. So staining basically increases the contrast and resolution of the bacteria by imparting them dye or color by coloring with stains and dyes. Preparation and fixation of bacterial smear. It is an important step in the identification of microorganisms in the microbial bacteriological laboratory. The microorganism can be isolated from various clinical samples such as urine, pus, sputum, body fluids, and CSF. The refractive index of the bacterial cell is similar to the refractive index of the medium, so they cannot be identified in an unstained preparation when seen under the light microscope. So staining is very necessary because it gives a better contrast to the bacterial cell wall by which we can see better under the microscope. How is the preparation of a smear done? We need glass slide, inoculation loop, culture media, normal saline, methylene blue stain, spirit lamp, filter paper, immersion oil, and microscope. So the procedure involves various steps, which includes first, glass slide is cleaned and made grease-free by wiping with the filter paper. Then the slide and the inoculation loop are heated over the flame to sterilize them. Mark the area on the slide where smear is to be made and put a drop of normal saline over it. Aseptically add the bacteria to the water, mixed bacteria in and spread. Air dry the smear. This will take some time. Then afterwards, flame the slide 2 or 3 times and allow the slide to cool. Now we are ready for staining. Now place the fixed smear on a staining loop or rack over a sink or other suitable receptacle. Stain slide with alkaline methylene blue for 1 to 1.5 minutes. Wash stain off the slide with water for few seconds. Blot slide dry with bibulous paper. Be careful not to rub the smear when drying the slide because this will remove the stained bacteria. Now the stain is ready to be examined under the oil immersion lens. So there are few precautions to be taken while preparing the smear. First, when heat fixing a smear, always make sure that the smear is on the top of the slide as you pass it through the flame. Second, bacteria growing on solid media tend to cling to each other and must be dispersed sufficiently by diluting with water. If this is not done, the smear will be too thick and uneven. Be careful not to use too much paste in making the smear as it is easy to ruin your results by using too many bacteria. Fourth, always wait until the slide is dry before heat fixing. Fifth, fixing smears with an open flame may create artifacts. Sixth, the inoculating loop must be relatively cool before inserting it into any broth as it will splatter the broth and suspend the bacteria into the air, which is infectious. Always flame the inoculating loop after using it and before setting it down. So, some of the frequently asked questions. First, what are the two purposes of heat fixation? Heat fixing kills the bacteria in the smear and bacteria gets firmly adheres the smear to the slide. Second, it allows the sample to more readily take up the stain. Second, what is the purpose of simple staining? Simple staining helps to increase the contrast and resolution of the bacteria by coloring with the stain and dye so that it can be easily observed under oil immersion lens. Why are basic dyes more successful in staining the bacteria than acidic dyes? So stains are basically the dyes. They carry either a positive charge, so basic dyes, basically carries the positive charge, or negative charge, which are carried by the acidic dyes. Bacteria, which are slightly negative charged, so they attract the basic dye. So that is why most of the stains in the laboratory are basic dyes. Fourth question, name three basic stains. Crystal violet, carbon fuchsin and methylene blue are the three basic stains.